Okay, so we've made it to exercise 10, which is skeletal muscle histology, or really what muscles look like under the microscope. And we had a little bit of time looking at these during the lab on histology, but this is going to dive in a lot more in depth. So the objectives should be to list the functions of your skeletal muscle, describe how it's organized, describe the structure of a muscle fiber and label it as a slide or a diagram. Describe the structure of the neuromuscular junction and label a slide or diagram. A very brief overview. We know from histology that there are 3 kinds of muscle tissue. There's the cardiac muscles, smooth muscles, and then skeletal muscles, which is what this lab is going to focus on. Skeletal muscles can shorten in length, and that produces movement in the body. So muscle tissue is excitable, just like nervous tissue, but it is the only tissue that is contractible. That's that shortening in length. Muscle tissue is attached to the skeleton directly or indirectly, and that can be like through tendons. And even though it's mostly made up of skeletal muscle tissue, there's going to be other connective tissue, nerves, and blood vessels associated with that tissue. Therefore, each muscle is considered an organ. As a whole, this skeletal muscle is going to play many roles and have many functions. One being to pull on bones to make movement of the skeletal joints, to help maintain posture, supporting and protecting soft tissues in the abdomen and pelvis, controlling entrances and exits of the digestive and urinary systems, producing heat to maintain internal body temperature, and serving as a reservoir for amino acids that can be used in times of poor nutrition. If we remember back from histology, we know that skeletal muscle is a voluntary muscle. So we can consciously control this muscle and consciously control many of these functions. When we look at muscle cells, there is a lot of very specialized vocabulary that is important for understanding what we are looking at. So with that unique set of vocabulary, we are going to spend several seconds on these slides with just vocab words and then pictures to sort of put them all together. So let's dive right in. The first vocab word we need is tendon. A tendon is this rope-like bundle of dense, regular connective tissue and is located on the end of muscles. It also can have collagen or other fibers, and it's going to attach the muscle to the bone. There is a fascicle. A fascicle is a bundle of muscle fibers that are found within the muscle. They're surrounded by the perimysium. A muscle fiber is a single individual muscle cell, and it's wrapped in a surrounding sheath of tissue called endomyosin. Endomyosin is a delicate connective tissue layer that is going to be around each individual muscle fiber or muscle cells. And it's really going to help it stick to other muscles, and it's going to be where there's some nerves and capillaries. We have that perimysium that we talked about with our fascicle. That perimysium is the connective tissue around the fascicle. It's going to have a lot of collagen and elastin. It also has nerves and blood vessels running through it. And then last but not least is this epimysium, which is a dense layer of connective tissue that has a lot of collagen fibers that is around the entire muscle. That tendon that we talked about, that thick bundle of tissue, is made up of that epimyosin, perimyosin, and endomyosin coming together at the end of the muscles and then attaching it to the bone, specifically to that periosteum layer, the outside tissue on the bone. So here's what we mean by those words. You have your tendon. That's this dense tissue here that is connecting to the bone. Here is like a big chunk of muscle, like in your arm. When we look at the outside edge of that big chunk of muscle, that's our epimyosin. Then we can see inside that there are these little rounded bundles inside of bundles inside of bundles. This next bigger bundle here, that is going to be your perimyosin. That's the connective tissue that is holding on to our fascicles. These little tiny bundles that you see here, those are your fascicles, which are groups of muscle fibers all stuck together. And then if we pull one of those out and look at one of these individual cells, that's your muscle fiber, and it is wrapped up in the endomyosin. So you've got your fiber wrapped up in the endomyosin. A bunch of fibers together make your fascicle wrapped up in the perimyosin. A bunch of fascicles all together wrapped up in the epimyosin makes up your muscle, which will connect to the bone by a tendon. All right, some more vocab words that we need. We need myofilament. That is going to be thick myosin filaments and thin actin filaments. That's what builds up individual muscle cells. We have myofibrils, which are these cylindrical organelles that are inside a muscle cell, and they're made up of those myofilaments, and they can be made of actin and myosin running from one end of the cell to the other. Again, muscle fiber is just the same thing as a muscle cell. You have a sarcomere, which is what is the functional subunit of your myofibril. So we're going to look at a tube that is going to be your myofibril, and we'll see that there is a little functioning unit that is your sarcomere. You'll also see something called A bands. A bands are going to have thick and thin filaments. Think A for A band, A for all myofilaments. And they're going to appear to be these darker, thicker banded regions. Then there is the I band. I band, think of the letter I as being a tall, skinny, thin letter. I bands are just made of thin filaments with actin. They are going to be found on either side of a Z disc. That Z disc is just the marking of what makes a sarcomere. So think of Z as like the end of the alphabet. It's the end and the beginning of a new sarcomere. So we have our myofilaments, that's actin and myosin. They're going to make up a myofibril. That myofibril is going to make up a muscle fiber. That muscle fiber is going to make up our muscle fascicle, and many muscle fascicles will make up the whole muscle. So when we think of muscles, a lot of times the sentence that gets used is it's a bundle inside of a bundle inside of a bundle. So you've got these filaments bundled together to make that myofibril. You've got your myofibril bundled up together to make a muscle fiber. That's the individual cell. You have muscle fibers bundled together to make fascicles. fascicles bundled together to make up your whole muscle. We also need a few more words like sarcoplasm. That is the special vocab word for the cytoplasm that is in a muscle cell. We have that Z disc, again, that's the end line of the sarcomere. We've got the sarcolemma. Sarcolemma is the special name we have for the cell membrane of a muscle cell. We have the sarcoplasmic reticulum, which is the smooth ER of our muscle cells. It's going to wrap around the myofibril, and at the end, it will have a terminal cisterna, which is going to contain calcium. And that's how we're able to release calcium when an impulse reaches into the T tubule or transverse tubule at the end of these sarcoplasmic reticulum. We also need neuromuscular junction. That's just where the axon terminal of a neuron rests up against the motor plate of a muscle. And the axon terminal, that's just the end part where we're going to release neurotransmitters. So again, we have our myofibrils. That is going to be like our actin and our myosin. And they are what make up a muscle fiber, the muscle cell. So we got a lot of our myofibrils coming together to make up the whole cell. And we can see here, there's our sarcolemma, this outside layer that is the cell wall. And inside are all of our filaments. When we look at it, we see this spread out green stuff here. That's your sarcoplasmic reticulum. That's the thing that's going to be holding on to that calcium. And when it is triggered to release it, it's where that is released. We've got this yellow bit, which are your T tubules. That is going to be like where that impulse that will tell the sarcoplasmic reticulum to release calcium is passed through the cell. Muscle cells have a lot of mitochondria in order to make ATP, because in order to move these filaments, these myofibrils across each other, we need ATP. You'll also see the many nuclei of our muscle cell. Here is that Z line. That is our sarcomere, our functional unit that makes up our muscle cell. Basically, we have a Z line here. It's like a zigzag ridge and a zigzag ridge here. And this is where that shortening, that contraction is happening. So it is where we're actually doing the work of a muscle cell. Again, we have the I band. The I band only has thin filaments or actin filaments. We have the A band, which is this darker area that has thick and thin filaments. And these will slide over the top or pass over the top of each other to make that muscle contraction. Again, here's what that looks like. This dark zigzaggy line, that is your Z line, making up your functional unit of your muscle cell, the sarcomere. Then we see this lighter stretch that runs like through our Z band here. That's your I filaments, your thin only filaments. In between this darker stained region, that is your A band, which has our thick and thin filaments. That's the all filaments. Then you have this line right here. That's your H zone. H zone has only thick filaments, only myosin. What's going to happen with the use of ATP is that we're able to slide these Z lines closer together, and that's the contraction of your muscle. If you need help visualizing what I just said to you, this crash course will walk you through how that happens. This video is in the D2L for you. I'm not going to play it now. You won't hear the sound, but this will walk you through exactly that shortening where those filaments slide past each other and how that all works. If my words and my diagram are not helping you to see it happen. Next, we need a few more words like motor end plate. That is the region on the muscle cell plasma membrane, right? That sarcolemma that is able to take in the acetylcholine neurotransmitter. So here's what that looks like. You've got your nerve coming right up to the sarcolemma. This axon of this nerve cell has these vesicles that are filled with acetylcholine, which is a neurotransmitter. Those vesicles are going to fuse with the membrane at the end of the axon. And when it does that, that acetylcholine is going to go into the synaptic cleft. Basically, it looks like the axon touches the sarcolemma, but they don't. There's a very small gap. Within that gap, the neurotransmitters move from the axon to the sarcolemma, that membrane that's on the outside of our muscle cells. There are going to be acetylcholine receptors on the sarcolemma that take in that neurotransmitter, which then tell that muscle to do the contraction, to let go of that calcium that is stored in the sarcoplasmic reticulum, and to move those Z lines closer together to make a muscle contraction. So, what are you going to be doing today? You should go through the lab resources, the lab study guide, take a look at the Quizlets that have pictures of the models of the muscle cell, of the sarcoplasmic reticulum, of the axon terminal, all of that, so that you feel comfortable. You should go through all of the information that is on the top hat. That's the lab manual book. So it has lots of different pictures and models to show you there. And then you should complete that work and then study for your quiz. And the quiz is open on the D2L. And as always, if you have any questions, please feel free to ask. But thanks for watching, and I hope you have a great day.